Our Capstone project focused on the virus filtration processes of diverse feed streams. Our group consists of Sarah Benson, Emma Rach, Matt Solarczyk, Arian Tsai, and Kendall Zerman. Our project sponsor was Sartorius, which has many products including virus filters to ensure high virus safety for patients. Virus filtration is an essential part of developing human or animal-derived biopharmaceuticals in order to ensure the drug safety. Failure to detect viral contamination can lead to serious consequences, mainly threatening patient safety and also potentially causing drug shortages and a loss of public confidence in drug manufacturing. Because of this, viral clearance is heavily regulated by the FDA, and a viral filtration step is necessary in the purification process. During viral filtration, small amounts of contaminants can cause membrane fouling or the premature blockage of the filter, which reduces product throughput and decreases the integrity of the filter. The use of a pre-filter prior to the virus filter has been shown to decrease aggregate formation and prevent filter clogging. The objective of our project is to investigate and quantify the effect of prefiltration on the throughput and flow decay of more diverse feed streams, as most prefiltration studies have mainly focused on monoclonal antibodies. The two feed streams that we used were bovine serum albumin, also known as BSA, and human serum immunoglobulin, or IgG. Our experiment design setup included two types of virus filters, the Virosart HF and the Virosart CPV, as well as the Virosart Max, which is our pre-filter. In a pressurized system, we ran our feed streams through the pre-filter and filter, using a scale and data collection software to obtain mass throughput and flow rates. We completed 5 runs using BSA, 2 control runs without a pre-filter, and 3 with a pre-filter. Our runs with a pre-filter were completed with 2 different ratios based on the surface areas of the filter to the pre-filter. 2 of them were done with a 1-to-1 ratio, and 1 of them was done with a 1-to-4 ratio. We then completed 6 runs with IgG, where 2 were done without the pre-filter and 4 were done with, where 2 of them had a 1-to-1 ratio, 1 had a 1-to-4 ratio, and 1 had a 1-to-12 ratio. From our experiments, we concluded that IgG shows better filtration compared to BSA when comparing the relative flow rate to mass throughput. Clinically, we know some feed streams may need a pre-filter more than others, while some may not need a filter at all. Our results indicate that the use of a pre-filter is dependent on the specific feed stream being used. We also conclude that additional experiments are needed to determine the performance differences between Virosart HF and CPV filters. It is important to note that due to budget and supply constraints, we were only able to perform 1 run for each experimental group. We would like to thank our Sartorius contacts Aaron Gladstone and Alex Schwartz, as well as Jean Gerber and our faculty advisor, Dr. Dan Hayes, for their support and guidance during this project.